So Dr. Clark is going to discuss early results, I think, correct, on his study looking at combination therapy of navigated laser and Lucentis.
So what I'm going to present today actually is not results, but sort of a study design that's we're in the process of initiating right now. We've actually sort of gone through all the regulatory hoops. It's been a long process, and I think this is a very interesting trial that we're initiating in a multi-center but investigator-sponsored way, evaluating combination anti-VEGF therapy with navigated laser. One of the interesting things we're talking about directed laser with the navigated laser in our trial that we'll talk about in a minute. We're only going to be treating microaneurysms. There's no grid laser. And so I think that's important as sort of an entry. But the other thing to keep in mind about our trial that we're starting is that I think this represents a very unique and interesting collaboration in terms of industry in the United States. This is a collaborative trial between 3 large retina practices diffusely geographically across the United States. And it's also a very unique collaboration in industry in that we've got Navilas as well as Genentech participating as collaborators in a very, very well-funded large clinical trial evaluating patients with DME. So I think, I think it really speaks highly of the company to be able to collaborate in this way to try to answer important questions as we move forward. But I think it also gives you a sense of how a major player in the market, Genentech, sees the technology that Navilas brings to the market. So with that introduction, we're going to talk about a phase 1/2 clinical trial that's an open-label 3-arm trial that's going to evaluate the safety and tolerability as well as the efficacy of ranibizumab in the U.S. dose, which is 0.3 milligrams in monthly treatment, which is really our label dosing. Our second arm is a treat and extend protocol that I'll explain in just a few minutes. And then the third group is going to be a treat and extend protocol with the combination, with the addition of navigated laser to treat microaneurysms when there's leakage in the macula. So our primary objective is to assess those endpoints of safety, tolerability, and efficacy of ranibizumab as well as laser in patients with DME. Again, we have 3 groups, and this is a very large investigator-sponsored trial. We've got 150 eyes that's going into this trial. We're going to have 3 groups. The first group is the monthly cohort, which is the on-label dosing for ranibizumab, 0.3 milligrams monthly. The T-REX, we've got this sort of dinosaur motif, which is a little bit hokey, but the T-REX is sort of an abbreviation for treat and extend. And so the T-REX cohort is and again, we're going to have a 2:2:1 randomization, but the T-REX cohort are patients that are treated with monthly treatment for 4 visits, and then based on treat and extend criteria that we'll show in a little bit. And that's one of the real struggles as we've moved forward with the investigators, is to actually figure out what the right paradigm is for treat and extend, because we really don't have a lot of guidance in evidence-based medicine to date to try to figure out actually how to do that. We do it in clinical practice, but we all do it a little bit differently. So we're trying to, at the same time, standardize treat and extend as well as standardized laser. And then finally, the Gila cohort, which is really most important to our discussion today, is essentially the treat and extend group plus guided laser only to microaneurysms when there's leakage in the macula. So again, we're going to...
What's the acronym for the first cohort?
Well, I guess that's...
Yeah. Well, I congratulate you. Actually have an acronym that makes sense. You can remember.
Yeah.
Treat and extend.
T-REX. That's right. That's right. So we need to come up with one. That's a good point. So we've got 150 eyes, 2 years. This is actually the largest investigator-sponsored trial that Genentech has funded since the launch of Lucentis in terms of their contribution to the trial. And of course, Navilas has made a very important and extensive contribution as well. We're going to enroll patients again in a 1:2:2 randomization scheme where we're putting 30 eyes into the monthly group, then 60 eyes in the treat and extend, and then 60 eyes into the treat and extend plus laser. Our primary endpoint is the mean change in ETDRS visual acuity out to 2 years. We're going to have a couple of different endpoints, which is again, it's an interesting, we're going to have a hard endpoint as well as what we call a biologic endpoint, which is when we have the maximum effect of the drug. We have a number of secondary endpoints as well. These will be things that we follow and we use to evaluate post-hoc analysis, adverse events, the total number of injections. This is actually quite an important secondary endpoint because we believe our clinical hypothesis is that we don't need to use ranibizumab monthly even without laser, but our hypothesis is that with navigated laser, we believe, based on our anecdotal experience as well as early data with a laser, that we'll be able to significantly decrease treatment burden as well. We're going to look at OCT central subfield thickness. We're going to look at 1-line and 3-line gainers. And we're going to look at a number of other anatomic outcomes associated with these patients. The major inclusion criteria is going to be a little bit more generous than what we typically see with diabetic macular edema clinical trials for FDA approval. These patients can be enrolled with relatively good vision. Our best corrected visual acuity can be between 20/25 and 23/20. Patients do have to have center-involved macular edema by OCT. Major exclusion criteria where we are not going to include patients with active proliferative disease. They can have received PRP in the past, but it needs to be quiescent prior to enrollment. We're going to exclude patients that have vitrectomy surgery. They can't have had any intraocular drug for at least 90 days, and we are excluding any macular laser because we think that's a significant confounding factor moving forward. So the T-REX cohort, again, this is the interesting part. So the monthly group is a group that makes pretty good sense. It's what we do in clinical practice when we're treating patients with anti-VEGF therapy. We do either monthly or fixed interval anti-VEGF therapy. For the T-REX cohort, patients are going to be treated with 0.3 milligrams at day 0, and then every month for a total of 4 visits. At that point, we're going to institute our treat and extend algorithm, and I'm sure we could probably fill the rest of our time tonight discussing treat and extend algorithms, how this one has some positives, it has some negatives. This has been kicked around for the better part of a year and a half with the folks from Navilas, the folks from Genentech, as well as my co-investigators, Dave Brown in Houston and Dave Boyer. And we've tried to come up with something that makes sense in terms of a real-world application of treat and extend in diabetes. So after 4 injections, if the patient has a central retinal thickness of less than 350 at week 12, we're going to move them into the extension phase, understanding that many of these patients, we're not going to get completely dry, but we hope to get their disease under good control. We're then going to essentially establish what's called a baseline retinal thickness, and we're going to establish a number for that patient. And then moving forward, that number will be our basis to make treatment decisions based on an algorithm we'll see moving forward. If the central retinal thickness is above 350 microns after 4 injections, then we'll continue to treat monthly, because I think most of us would agree that it's certainly a cutoff at 350. It suggests active disease. That number has been one that's been of significant debate within our study group, but we've settled on the number 350 because we want to actually be able to extend a significant number of patients to see if we can decrease treatment burden. The GILA cohort is very similar. Again, they're going to get a total of 4 injections, and they're going to follow the same treat and extend protocol. The difference is these patients are going to receive navigated laser via the Navilas platform. We are going to treat these patients at month 2. So they're going to have a little bit of anti-VEGF therapy, and then after an initial injection to try to decrease the macular edema, we're then going to laser them at the second visit to try to treat microangiomas. And then we're actually going to obtain angiograms quarterly. And if there is leakage on the angiogram, then we will perform repeat navigated laser treatment only to microaneurysms, no focal laser. So again, these patients will not receive any grid laser during the course of the trial. The treatment interval determination, this is where things get complicated, and we're just getting started. We actually got all the regulatory approval done, and the press release was actually released this week about this important investigator-sponsored trial. So we're in the process of training ourselves and training our co-investigators. But essentially, the basic concept is if an eye is relatively stable, we're going to extend the interval. If the patient is not doing particularly well, we're going to maintain the interval at its current rate. So if they're at a 6-week interval, we're going to leave them at 6 weeks. And then, of course, if patients are doing quite poorly, we're going to reduce the interval by 2 weeks. Here's sort of a schematic of how that's going to work. So we're going to, again, establish a retinal thickness baseline for these patients. It has to be somewhere less than 350 microns, and for that person, they're going to get a number. And as long as that patient stays within 10% of that number, as we increase the treatment interval, we're going to continue to increase the treatment interval. If they fall outside that 10%, if they are either significantly thinner or if there is a slight increase between 10 and 20%, then we'll leave the interval the same with the idea that we don't believe the disease state is stable and we want to sort of see how things are going. And then finally, if we see significant thickening better than 20% of their baseline, then we'll reduce the treatment burden. You know, these numbers seem, the 350 cutoff is one that's been particularly controversial in our study group, and some of these numbers have been difficult as well, because I think for treat and extend, many of us think about that paradigm within the context of age-related macular degeneration. We worry about the impact of a recurrence in terms of the long-term visual outcome for patients. In this disease process, however, we feel like that small recurrences are probably not as much of a concern, and we'll be able to recover visual acuity. So we've tried to sort of loosen our criteria to some degree in hopes that we can demonstrate decreased treatment burden from these patients. We are establishing as well a secondary as well as tertiary baseline. A secondary baseline will be established if the study eye has had 3 consecutive visits with at least a 20% reduction. We'll then re-zero them and then move forward. And then a tertiary baseline will be established if the study eye has 3 consecutive visits with a 20% reduction compared to the previous baseline, and there is a less than 50 micron variability between those visits. This is very complicated. We recognize that it's a very complicated sort of paradigm and protocol, but we're hoping that what we can do in this trial is start to explore the idea of treat and extend to try to organize and more standardize how we're going to manage these patients going forward, because we don't believe that monthly treatment long term is a good outcome for these folks. Finally, we'll have a number of endpoints. We'll have a hard endpoint at 1 year and 2 years. We'll also have a biologic endpoint, which will occur 4 weeks after their last injection, and we'll see if there's a difference between the two. You know, based on when you look at clinical trial design, it's important to look when their last intervention was, and so perhaps it may not make a difference, but we'll see if there is a difference at 2 years between what we would call a hard endpoint and a biologic endpoint. Finally, through the generosity of Genentech, we'll have access to fellow eye treatment in these patients. So any patient that develops DME after they're initiated in the trial, we can enroll both eyes in the study protocol. But if a patient develops DME after initiation of the trial, we'll have drug available. I want to recognize my co-investigators. And again, this is a process that's been going on now. This is a very difficult sort of idea to get your hands around, and a very difficult idea to actually get the co-constituents of in terms of laser companies and the pharmaceutical companies to buy into, because it's a difficult idea. But after almost 2 years of working on this protocol, our group has been able to finally get this protocol done, and we're sort of at the advent of getting it off the ground. So that's Dave Brown in Houston, Texas, and Dave Boyer in Beverly Hills. So it's a complicated trial. We're hoping to learn quite a bit about DME management, both in terms of treat and extend, as well as the role that navigated laser plays. And our hypothesis is that navigated laser is going to offer us a substantial decrease in treatment burden. And so our expectation going in is that we'll see incremental decreases in treatment burden moving forward. Thank you very much.
So when do you expect to start enrolling?
So we will start enrolling within the next couple of weeks. We're due to get drug from Genentech sometime within the next week to 2 weeks. We have gotten a full go-ahead. We've gotten our IND is in place. We've got approval by all the regulatory bodies.
So when you did your power calculations, you assumed a certain difference in the visions in the 3 groups. I might know more than I'm leading on, but maybe you can share with the group what you're thinking. You know, when you do a clinical trial,
Right.
you have to have some
Right.
presumed
Yeah.
effect. What were your thoughts?
Well, so we probably need, in terms of, to show any type of superiority between treatment groups, our trial's not big enough. We're looking for signals. We're really, that's why we added in as a key secondary outcome a non-inferiority analysis, because we believe at the end of the day that what we're going to be looking for in terms of visual acuity is non-inferiority. And then as a, we think it's likely that what we'll see is not a lot of difference in visual acuity, non-inferiority between the treatment groups, but a significant difference in treatment burden. A lot of what happens when you're doing investigator-sponsored trials in collaboration with industry is that you're limited by your funding. And so, again, this is we're not powered large enough to show superiority between treatment groups, but with that in mind, we've been able to fund the largest IST that at least Genentech's being involved in. So we're very pleased about being able to do that in collaboration with them and with Navilas. And what we're hopeful is that using our non-inferiority analysis, we'll be able to show no differences in efficacy, but with significant decreases in treatment burden.
Right. So, you know, a lot of people keep saying we don't really know what the systemic issues are with anti-VEGF drugs, but actually we do. And if you look at the FDA label for Lucentis, it's in there that there is an excess incidence.
Oh, you know, there's no question that there is an incremental increase in serious adverse events and cardiovascular adverse events every year that RISE and RIDE went along. That's one of the reasons why you know, there's this talk that EYLEA is likely to get approval after 12-month data. I'm surprised with that because of safety. When you see the incremental increase in adverse events in the diabetic population, I think long-term data on these patients is important. So I think anything we can do in patients that have an increased risk of cardiovascular events to decrease their anti-VEGF load is critical.
Right. Because there will be no more placebo-controlled trials, so you're never going to have that risk data. But it was done in the initial licensing studies for Lucentis. Very interesting.
Great, thank you.
It works out well.